Yeah, so I'll be talking about this work and uh, that we did, like I did with Shakti and Sitabra. And I'm really grateful to both the institutes, Indian Institute of Technology, Gandhinagar, and Institute of Mathematical Sciences, because of whom I could do this work. And from the topic, I think you can figure out that I'm interested in decoherence rather than coherence, which is generally an area of keen interest. Yeah, and secondly, the non-linearity of interactions. Right. So, a brain is a dynamical system. We all know, and inherent non-linearity in the brain allows it to do the plethora of functions that it does for us. And of course, these dynamics are shaped by the topology of the brain or the underlying neural architecture. There are three scales of interest when we talk about dynamics in the brain: microscale, mesoscale, and macroscale. And in terms of spatial and temporal separation, when we say about microscale, we are really talking about microns in space and milliseconds, of the order of milliseconds in time scales. At the mesoscopic scales, we increase the spatial resolution to millimeters and centimeters, and temporal to milliseconds to seconds, whereas when we are talking about macro scale, we are really talking in terms of centimeters and seconds. So I think you can appreciate that at microscopic scale, we are talking about neuron dynamics, and the ion channels and gates, ion gates within the neurons. But at the mesoscopic scale, we are talking about brain areas or ensembles of neurons, whereas at the macroscopic scale, we are talking about the whole brain dynamics. And to just give you a sneak peek into what do these dynamics look like, so just imagine the brain is made up of different brain regions, here shown in 3 different colors, and each of these brain regions have their own networks of neurons that interact with each other, which are shown with these smaller circles. So at the level of neurons, we are talking about this conductance based model like Hodgkin and Huxley, and what we are simulating at these neuron scale or micro scale are these action potentials. So there is so they are spikes basically in the membrane potential of the neurons. And at the mesoscopic scale, which is at the level of neuron ensembles or brain regions, we are looking at the oscillations of these entities, and they generally have 2 populations because neurons could be excitatory and inhibitory. So these 2 curves here are each depicting an excitatory subpopulation and inhibitory subpopulation. I'll get to them in detail a bit later. Right, and so at the mesoscopic scale, what happens is, so this is, you might have figured by now that I'm interested in mesoscopic scale. So it's intermediate between microscopic and macroscopic. So hence its dynamics are affected by the other 2 scales, by the processes at other 2 scales, and that leads to emergence of complex spatiotemporal patterns. And the dynamics at this scale generally comprises of oscillations of different frequencies. And what we measure in EEG are generally the mesoscopic dynamics, whereas if you had LFP, you are really looking at the microscopic dynamics. And this all enables us to use the very rich mathematical and statistical modeling theory to capture the emergent dynamics. And one such class of models are neural mass models. So neural mass models are just low dimensional representations that aim to describe the coarse grained activity of large populations of neurons and synapses between them. So you no longer worry about neuron level connectivity or the dynamics within the neuron, what are the time constants, the conductances and the gating variables, etc. So you just care about what are the mean activities or the coarse grained description of activities of large populations of neurons. And neural mass models have several advantages, but 2 main of them are that they are simple, so they do not incorporate these neuron level structure or details of connectivity, and hence have very fewer parameters, and that essentially also makes it a low cost computational model. So if you were to let's say study a brain process or reproduce some of the dynamics of the brain, and if you were to look at individual neurons, you have 100 billion neurons, so it's nearly impossible to simulate all of them. And so neural mass models are a smart way to be able to reproduce the processes such as seizures or sleep that you're interested in with fewer parameters and do it more computationally feasibly. And one thing which is very important is that neural mass models are assumptions of, so they assume that there is there is arbitrary they are arbitrary discretizations of actual brain. So they assume existence of local dynamics. So they assume the brain is made up of these different subunit different units, each of them they have their own dynamics. But then actually what happens is the brain tissue is a continuum. There are no like separate entities, right? Brain tissue is a continuum. And when we talk about like networks of these neural masses, we are making another assumption, right? Because true network only exists at micro scale. So neuron talks to another neuron. So true network exists there. But when we talk about networks of neural masses, we are making another assumption. So this is one of the feedback I got in my PhD thesis dissertation. One of the reviewers said, you should write this in your thesis. So I always put it up in my slides. Okay. And then I think this audience already knows a number of classic neural mass models. And of course, my talk would be focusing on the Wilson-Cowan neural mass models. But then there are others like by Lopez de Silva, Freeman, Jansen and Ritz, Stefanescu et al. And Wending et al. And each of them are neural mass models, but they are specialized for modeling different different brain process. For example, these two are for modeling the EEG like activity, whereas Jansen and Ritz again is also for EEG, and it's a model of cortical columns, whereas by Wendling you see it's a model for epileptiform activity. So these are specialized, some of them for EEG, some of them for reproducing the epilepsy like activity. Right, and talking about neural mass model, it's unfair to miss out the Kuramoto model. So it's a very classic, elegant model proposed almost 50 years back by Yoshiki Kuramoto. So he proposed a very simple yet elegant phase oscillator model. So a phase of ith oscillator evolves with this differential equation. So at d theta by dt is sum of the intrinsic angular frequency and the coupling constant k and the summation of the contribution from the rest of the oscillators that it couples to with this coupling strength. And this is just a difference of sign of the difference between the phases of ith oscillator and rest of the oscillators it couples to. And so right, so the in a system of Kuramoto oscillators, the order parameter that captures the phase coherence or the alignment of all these oscillators is this parameter R. So you can see it's expressed like this. So when all the thetas are equal, which means they are all phase synchronized, you can see that R would be 1, and psi, which is the mean phase, would be equal to the theta to which all of these are equal to. And here's a very nice diagram that shows you 6 different Kuramoto oscillators which are each coupled to each other with this coupling strength K, and the red dot at the boundaries is telling you the instantaneous phase, and the arrows within the circle is telling you what is the intrinsic frequency of these oscillators. So you can see that they are all different. And now as you increase the coupling strength here from k0 to 0.01 to 1, you can see that vector r, which is captures the phase coherence, it increases in its radius. So it becomes more and more coherent, the system becomes more and more coherent as you increase the coupling strength. and this is also clear in this diagram R versus K, where you see a transition to synchronization as the coupling is increased. So this model is used typically to study synchronization of coupled biological oscillators. But then this model has several limitations, not to say that this model is any less in terms of when it comes to studying synchronization dynamics in brain, but then it has no amplitude dynamics, it has only phase. There is no distinction between inhibitory and excitatory subpopulations. It has limited biological interpretability, so it doesn't have any plugins like physiologically inspired parameters in it, so that you can say, okay, this is inhibitory population, this is excitatory population, these are the it doesn't have any biological interpretability. That's the main point. And it has sometimes a very limited capacity to model multi-stability, oscillation onset, or bistability. So today in this talk, as you might have guessed, I am interested in studying transition to decoherence with increased coupling. So in our brain, it's not always that when the interaction strength is high that we get synchronization. The opposite things also happens. For example, under the influence of under sleep, REM like sleep, and under the influence of psychedelics, there are cases when there is increased interaction, but there is no global synchronization between the brain areas. So we really think that the Kuramoto like paradigm would fall short in reproducing such dynamics, and therefore we turn our heads to neural mass model, and specifically Wilson-Cowan model, that first of all takes care of all these limitations, and will potentially see how it reproduces this phenomena. So I again think this audience knows about the Wilson-Cowan model. So the Wilson-Cowan model assumes that a single brain area or the ensemble of neuron, so it takes care of activity of mean populations of excitatory pool of neuron and inhibitory pool of neuron. So this is a single unit of Wilson-Cowan oscillator. So U1 is like excitatory subpopulation pool of first unit, and U2 is the excitatory subpopulation unit of subpopulation of unit 2, and each of these units receive external stimulus IU2, IU1, and the neurons within the subpopulations are coupling to each other. Like here, there is also coupling between inhibitory and excitatory subpopulations, and between the two units, the coupling strength is depicted here by W. So each of these 4 populations are interconnected. So what does Wilson-Cowan model say? That the dynamics of U and V populations evolves with time with these equations. So this is the self term, and this is the term that takes care of interactions with the other units or Wilson-Cowan units or nodes or oscillators. So kappa U and SU, these are both, as you can say, they are sigmoidal functions. So you can see this term RU, UI and RU, VI, they kind of control the effect of this coefficient on the gain or the sigmoid response function here. So if RU and RV were 0, let's say, then kappa U, which is generally 1, I'll tell you now. So then you'll have full gain, full effect of these sigmoids, but when they are non-zero, it kind of reduces these coefficients. And then so SU and SV, which are the sigmoidal functions, they take these as inputs, and the inputs to each of these units is sum of 4 different terms. Of course, the external current like here, and so this is the term that takes care of coupling within the neurons in the excitatory subpopulation pool, CUU, UI, and then this is between excitatory and inhibitory, CUV, VI, and this is like sum of interactions from all other units it connects to. So, it is summation over j, right, and similarly for V in. So, this is the whole model, and as you can see there are number of parameters here, around 12, and AU, theta u, AV, theta v just take care of slope and thresholds for the sigmoid function. These are the internal coupling parameters. RU and RV are the refractoriness parameters. These are time constants, and IU is generally set to 1.25 and IV is 0. So, these parameters are taken from the original 1972 Wilson-Cowan paper, so that a single Wilson-Cowan unit is in the oscillatory regime. Now, let me talk a bit about the properties of the Wilson-Cowan model before moving on to the decoherence phenomena that we are interested in. So, as you can see that the model equations incorporate nonlinearities and lags and two different populations. The model equations can be used to calculate steady state levels of cortical activity for various levels of stimulation. So, here is the diagram that shows you as a function of IU, which is the input current to the uth population, and V, which is the mean activity of the inhibitory population. And if you plot max V and min V, there is a region here for this range of IU that the max and min do not coincide, which means that it is an oscillatory regime. Whereas here you see there is 0 amplitude. So, the oscillator is in amplitude death range regime, or here amplitude death, and here it is oscillator death. So, there is no oscillation, but there is finite amplitude. So, the parameters that I had shown you in the previous slide are such that we are in this oscillatory regime. Now, another interesting property of the Wilson-Cowan model is that it could be used to reproduce signals that look like EEG. So, here is the time series of the U population, sorry, there must be time here, and this is from one of the nodes of, so when you simulate the dynamics of macaque brain network, simulating each node as a Wilson-Cowan oscillator, so this is just a time series of one of the node that is reminiscent of a typical EEG pattern, and variation of coupling strengths leads to vast repertoire of collective dynamics. So, you can see here U1 and U2 are shown as a, again I have cut out time for some reason. So, you can see that as the this W, which is the coupling strength between the Wilson-Cowan units, is increased, you go from seeing an exact synchronization. So, both the oscillators are phase synchronized here. Here it is quasi periodic dynamics, and here this is anti-phase. So, both the oscillators are in opposite phase with each other, and this is inhomogeneous in phase oscillation. So, they have different amplitudes, but they are in phase. So, just by varying one parameter, you see a range of collective dynamics that this model could produce. Now, another, so Wilson-Cowan model is a very versatile model, and we have also used it in another work that we did to simulate visually evoked epileptic seizures. So, what I have shown you here is that for a constant input IU current, you see that these oscillators are each not in phase, they are in different parts of this unit circle. But when we give this periodic stimulus, of course with certain frequency, we see that all these oscillators are phase synchronized, and this is depicted as a time series here. So, before this perforated line there is constant stimulus, and after that there is a periodic stimulus, and you can see that because of this periodic stimulus they have synchronized. Yep. Yes. So, this is like, yeah, it depicts global synchronization, which is reminiscent of visually evoked epileptiform activity. So, now I think you have a fair idea of what basic Wilson-Cowan equations are, dynamics are, how it can be used for simulating different brain processes.
So does it synchronize to the same frequency of the driving signal?
Yeah, that's a very good question. Um, these Wilson, each of these Wilson-Cowan units have their own intrinsic frequency. So the driving frequency has to be close to that for this pattern to arise. If it is far from that intrinsic frequency, you won't get this synchronization. So this is kind of a resonance.
How sensitive it is?
But yeah, there's a range of frequencies near the intrinsic frequency for which we see this resonance coherence like phenomena, but beyond that it's not there. Right. So coming back to the original phenomena that we are interested in, which is strong interactions between brain areas can be associated with the loss of synchrony. So the objectives of the work that I'll be talking about today is to first of all see how Wilson-Cowan and the Kuramoto like phase oscillators pan out in reproducing this process, and then what is the role of network topology in transition to coherence, and then the role of model nonlinearity and refractoriness in such transitions. Right. So first we compare the dynamics of Kuramoto phase oscillators with the Wilson-Cowan oscillators. So you can see here again the same diagram. I've just shown you three different dispersions. So the frequencies of the Kuramoto oscillators are chosen from a distribution, and this just depicts whether how spread out is that distribution. So for low dispersion, you see the transition to synchronization is very quick. So even for very small values of k, we see a transition to synchronization, whereas for medium and high dispersion, it is a bit slower. And similarly for the Wilson-Cowan nodes, and here for fairness of comparison, we also take heterogeneous oscillators, whereas the previous works that I've shown you had assumed homogeneous oscillators. Right. So it's like sigma. It's basically standard deviation of a distribution from which we are picking up these intrinsic frequencies. So yeah, you have already seen these patterns before: phase synchronization, quasi-periodicity, and anti-phase. So what is similar with the Kuramoto oscillators is that we also see a transition to synchronization at very small coupling strengths. So although this is very compressed for you to see, but even with very, very small w's, 0.01, we see a transition to synchronization here. But what's not similar is apparent in the next few slides. So we see a transition to synchronization, and here I'm using a different order parameter which is zeta for Wilson-Cowan nodes. So when it is 1, it means that the oscillators are synchronized. When it is not 1, it means the oscillators are not synchronized. And we also see there is a similar transition to frequency synchronization. So you see that close to like between 0 and 0.1 roughly, right? There is a transition to synchronization in phases, but there is also a transition to synchronization in terms of frequencies. So you see that initially at w equals to 0, each of these oscillators have their own intrinsic frequency, but as w is increased, these frequencies keep merging with each other until and unless there is a single emergent frequency at sufficiently high w. Right, and yep, so that's there, and this is also clear in this diagram where we have just so there are just 10 oscillators, but here we have also varied n, and we have seen as the w is increased, which is along this axis, sigma f becomes 0, and when sigma f is 0, which means that all the frequencies are same and the system is frequency synchronized. But what's different from the Kuramoto oscillators is that at higher couplings, which is around 3.5 and 4, we see a transition to desynchronization here, right? And this is also the case for the frequency case. So here sigma f goes to non-zero values. So there is a transition to desynchronization at increased coupling, and this is the behavior that could not be reproduced by a simple Kuramoto like oscillator. So since we were looking at the heterogeneous Wilson-Cowan nodes, we looked at several collective dynamics. It's always interesting to explore them. So as we are increasing the coupling strength here, W2, 470 and 200, you're again seeing different sort of collective dynamics. This is inhomogeneous in phase synchronization 1, this is same, but 2, because they are both phase synchronized but the dynamics look different. And this is quasi periodicity, and this is inhomogeneous out of phase synchronization. So by inhomogeneous, I always mean that the amplitudes are different for each of these oscillators, and out of phase, which means the phases all are distributed, they are not synchronized. What we also see is, especially for the case of the heterogeneous oscillators, is if I just show you a parameter space diagram of n versus w and show you the number of nodes that are oscillating, right. So you see in the blue scale here, in the blue color here, the number of oscillating nodes decreases. So there is a huge patch in the parameter space diagram where we see things like this, where none of the oscillators are oscillating basically. So this is like an oscillator death scenario, and they are spread out because they are all inhomogeneous, they are not similar to each other. But we also see this sort of greenish range regime here, where a fraction of oscillators or nodes are oscillating. So you see just the 2 of them out of, I think, 25 or 30 are oscillating. So we call this state as chimera like state, where few of the parts of the system has one state while the rest of them have other state. Next thing is we look at the parameter space diagram again, but then we look at what is the dispersion in phases and the frequencies. So this diagram captures all the collective dynamics that we can see from this inhomogeneous Wilson-Cowan network of oscillators. So here there is IIS 1, inhomogeneous in phase synchronization 1, then there is inhomogeneous steady state, which means all the oscillators are not oscillating, they are just stuck in oscillator death, and then there is chimera pattern here, and then there is a huge range of inhomogeneous out of phase inhomogeneous out of phase state, and then there is IIS 2, inhomogeneous in phase part 2, and this is the corresponding frequency synchronization. And I want you to pay attention to this white line here, which is a peculiar thing that we observed. So generally when we observe phase synchronization, we also simultaneously see frequency synchronization, but at the boundary here, at the boundary between IOS and IIS2, we see that there is a sudden jump in the frequency synchronization. So they are not frequency synchronized here, but the phase synchronization, which is shown by this zeta value here, it is sufficiently high. So we have some phase synchronization but absolutely no frequency synchronization. So that's a phenomena of which is basically the discordance between phase and frequency synchronization that is seen in homogeneous Wilson-Cowan coupled oscillators. Now coming back to the network topologies, so we did some simulations on the macaque brain networks, and here's a yes, sorry.
Chimera state.
Yeah.
I might be very wrong here. So is the chimera state playing a role in this frequency switching?
That's a good point. Okay, so chimera state is somewhere here, whereas this frequency switching happens between IOS and IIS2. So it's likely that it doesn't play a role. Yeah. Right, and so this is adjacency matrix of the macaque brain network. It's a directed modular network of cortical and subcortical brain areas. So yellow means 1 and purple means 0, and here's just a depiction of the brain networks, and the node sizes here are depictive of the degrees of the nodes. Right. So what happens when we model each of the nodes in the macaque brain networks as Kuramoto oscillators versus as WC oscillators? So I'll show you 3 different versions of the network. One is macaque brain network, and then there is a network which is just a randomized version of macaque brain network, and then there is Erzos-Renai network, which just takes in the number of nodes and connections as macaque network, but it's a random network. Right. So this is what you see when you model each of these nodes in the brain networks as Kuramoto oscillators. So irrespective of which topology your network has, you see this unavoidable transition to synchronization as the coupling K is increased. Whereas if you have them as modeled as WC oscillators, you see that only for the random, completely random network, you see some transition to phase synchronization and then desynchronization, whereas for the other two networks, the macaque network and the randomized macaque network, you don't see any transition to synchronization. So which likely indicates that the real human brain topology or the macaque topology is somehow structured to avoid this global phase synchronization, as it would represent a pathological brain state like an epilepsy. And then we tried more like to break this, try to break this synchronization that we always see for the Kuramoto oscillators. We generated 4 different surrogate networks. So this one is normal macaque network, and this is degree preserved, module preserved version of the macaque network, and this is just the degree preserved network, and then there is module size preserved network, and then there is Erzos-Renai version of the macaque network. So you see, it didn't help for the Kuramoto oscillators. We always see this transition to synchronization, but for modeling the nodes as WC oscillators, we see that for the 2 most randomized version of the networks, we see this transition to synchronization, whereas there is no such synchronization for the other 3 networks, which again indicates that there is something about the real networks, that they are resistant to this large scale synchronization, which could be pathological. Yeah. And now we try to debug, like dig in deep, what is the likely cause of this transition to decoherence. And for that we did 2 things. First is we looked at the sigmoidal function. So this is the equation that I have already shown you before. So you can see that the sigmoidal function, which takes in this another sum of inputs as the total inputs. So we treat this coupling strength as a perturbation around the remaining terms. So the remaining terms we call it as x naught and delta x as w into u2 minus v2, and we do that for all the 4 equations. So we take a simple like network of 2 oscillators here, and then we expand the sigmoid function around this x naught and just retain the linear term. So we get rid of all the nonlinear terms. So this is just to see if we have a sigmoidal function in which just the linear contribution is there, do we get rid of the transition to desynchrony that we see. So there's the full model and the reduced model. So the full model you can, as expected, you see this transition to desynchronization at the usual place at around 3 or 4, W equals to 3 or 4. But in the reduced model, you see that you have avoided that transition to desynchronization, which means that the nonlinear terms in the sigmoid function play a role in transition to desynchrony. But then that is not avoided for a long range of W. So if I had shown you a larger range, it ultimately goes to desynchronization. Yeah, and another point that I forgot to mention before is that this transition to desynchronization is not trivial oscillatory death kind of situation. It is, the dynamics are there, all the nodes are oscillating, it's that just that they are not desynchronized.
Did you also check like which order term like contributes? Like, let's say
Yeah,
the
I
first
think
order,
we don't didn't do
second
that.
order, third
Yes,
order, what, like,
right. That's a good point. We didn't end up doing that. We just looked at linear term. And the second thing that we tried doing here is we looked at the role of refractoriness. So we all know that like each of the population in the brain is not always ready to fire. After firing, there is a period in which the neurons are refractory, and irrespective of however amount of stimulus they get, they can't fire. So that is captured by this term RU in these equations. So we looked at what would happen if the refractoriness wasn't there versus when it was there. So till now, whatever I've shown you, the refractoriness was there always. RU/RV was 1. Now look at, so this is what you would get. So for a small range of W here, you would see the synchronization order parameter is 1, but then after a certain point it decreases because of decoherence, and then there is oscillator death, and these different dynamics are presented here in terms of limit cycles. So you see that here it is synchronized, exactly synchronized. Here there is quasi periodicity and then antiphase and then oscillator death here. Now when there is no refractoriness, which would amount to just this term vanishing and just kappa u SU remaining, what we see is that we still see a small range of synchronization and then a transition to desynchronization. But what we see is that this range over which we got the synchronization has been shrunk. We just see a synchronization up to a smaller value of W here as compared to this case, which means that again indicates that the refractoriness is somehow controlling the overall gain that these populations have at each point of time. So, and because of that, the transition to desynchronization is further put off here, whereas not here, right? And with this, I think yes. Yeah. Oh, even higher values.
Are we busy with these?
If we go to higher values. Yeah, so we didn't find anything interesting beyond that. It was just desynchronized for a long value W before it finally went into OD state. Sorry?
You can sort of say that it's an expanded version of it, like RURB's plot is like an expanded version of the RURB's physical plot.
Yeah, you can. Sorry.
So, sorry, hold on. Like RURB equal to 0 is an expanded version of RURB equal to 1.
I don't know if you can say that because even the dynamics are pretty different. If you see here that the amplitudes have considerably increased, and these are winding number 2 limit cycles, and exactly because of that we had to switch to a different order parameter here, which is based on Hilbert transformation, because the other parameters like Kuramoto order parameter or the zeta didn't work for winding number 2 limit cycles. So it is entirely different sort of dynamical regime that we enter once we put this, But here our aim was to just look at what is the role of presence of refractoriness versus the absence, rather than, although it's an interesting problem and I think it could be a research problem of its own to look at the absence of refractoriness. Right. With that, I think I'll just conclude. So WC models show a transition to decoherence at strong couplings. Phase and frequency synchronization could be discordant. There is an existence of chimera states in networks of WC nodes. Network connection topology manifests itself in WC oscillator modeling versus in Kuramoto oscillator modeling. And absolute refractory period plays a role in fundamental nonlinearity in WC model. So that I'd like to end and take your questions. Thank you.
Thank you for the very nice talk. So we have more time for questions. So do you have an intuitive explanation as to why increasing coupling?
Yeah, it's killing the synchronization. That's what is happening. I think with increased coupling, you see the nonlinear terms become more dominant.
Yes.
Right.
How is it affecting? Well, generally, is it more coupling means?
Yeah. That's what is the general perception. We increase coupling, we increase synchronization. But the brain is a super complex thing. We see the reverse too, and we thought, how could we study and reproduce that? And Wilson-Cowan model gave us that, but we have just like some ideas about how it's happening. One thing is that, as we saw in the linear expansion of until just the linear term, we could see that the transition to desynchronization is pushed to higher Ws, even higher Ws. So we think that inclusion of like W with the nonlinear terms in the expansion plays a role in desynchronization. So that's our understanding, but it requires more like an analytical argument. I don't think we have.
No, actually, I think Namali was asking this is hand-waving argument. I think we have a semi-analytical argument, but we don't have a hand-waving argument. Like, easily explain to you.
Yeah, nonlinearity can do a lot of interesting stuff. Right? But I'm just curious, like, how is nonlinearity playing a role over here?
Yeah.
So does the transition point, like where synchronization and desynchronization happens, also depends on the internal coupling parameters CV, CV? This seems like Kuramoto has like one interaction parameter, and that has been sort of split into multiple interaction parameter in the other model.
So that's one thing that we didn't look at, but so I told you we are having heterogeneous Wilson-Cowan oscillators. So we are generating this heterogeneity by varying the internal coupling parameters. So our oscillators are different from each other just because their internal coupling strengths are different. They are being chosen from a distribution. But then if these internal coupling parameters have a direct impact on the point at which transition and desynchronization occur, I don't think we have a direct answer for that. So these oscillators are heterogeneous, and irrespective of the number of heterogeneous trials we make, the transition seems to occur at the same point. Transition to synchronization and desynchronization.
So actually, I do remember at some point we were looking at what would happen if we change the extent of radiation of the internal coupling.
Yeah. Right.
You also need to keep it in the oscillating regime. So if
Yeah,
you
yeah,
change it too
yeah,
much, it goes out of the oscillating.
Right, right. Yeah, we cannot vary too much, right? Yeah.
So, like varying the refractory period, refractoriness plays a role in decoherence.
It plays a role in?
Decoherence.
Yes.
So, like, is there like increasing it or any other parameter would also like make decoherence occur faster at smaller coupling strengths.
Yeah, so that, I think that's what happens here. So the transition to decoherence occurs earlier here at smaller w, if that's what you're asking. So do you mean if it can be
I mean, like
pushed?
this occurs fast, decoherence occurs faster, but
Yeah.
the oscillatory death happens at a very late.
Yeah.
So like I'm trying, if there any parameter that limits this distance to like minimizes the idea of decoherence occurs faster, but also oscillatory death occurs faster.
Yeah, that's a good question, but unfortunately we don't have an answer. We were interested in just looking at this and how is it manipulated by nonlinear techniques.
Like the decoherence occurs at smaller couplings.
Yes.
But the oscillator death happens later.
Yeah.
So is there any parameter that controls this oscillator that even occurs faster, despite there is decoherence of small couplings?
So sometimes I use one like oscillators, but since because of these internal couplings and there are so many parameters, how do you make sure that the behavior that you see is actually robust enough to variations of, because in Wilson-Cowan's paper you change a little bit and then you see these like very like this particular phase where there are double hysteresis loops and very complicated patterns and stuff.
Right.
So
Right. That's right. So we basically like we choose these parameters unstated otherwise always, and they are for so I showed you a diagram in which there was amplitude death, oscillator death, and limit cycle regime, oscillatory regime. So these parameter sets are to have oscillations, to always have oscillation, not have amplitude and oscillator death. But of course, we do vary the internal coupling strengths for getting heterogeneous oscillators, but we don't vary anything else in these parameters. And we did, sorry, no, I was saying we did vary the external stimuli a bit in that range. So, like so that we are still in the oscillatory regime, like we can vary it from here to here. So it's robust within that.
I was just going to say that actually the internal coupling parameters are in some sense the most important parameter, because the other parameters connect to like the steepness of the sigmoidal function and the time scale and so on. So the four C parameters are the most important ones.
Yeah, actually that does remind me, we also varied the sigmoidal parameters a bit, these AU and theta U, and for a fair range the dynamics were reproducible.